Next generation sequencing, or NGS, also called high throughput sequencing, is a popular method to generate many types of omics data. And it's a very exciting trend that started about 2010, when people started realizing that you can analyze the complexity of subcellular biological processes using one type of a machine, which is this next generation sequencer. So first of all, OM means collection, and a comprehensive collection of something. So we can think about phenomics, how we can analyze a collection of phenotypes. For example, when we look at this image of the cell culture, there's quite a number of things that we can categorize. It's cell size, morphology, maybe the tissue that it came from, maybe different kinds of variation that we can see here with the size of the nuclei. So together, all of these parameters could be referred to as phenomics. Then we obviously have what's called genomics. And so the genomic revolution that we are experiencing today, largely because of the low sequencing costs, refers to genomic variation. Genomic variation could be single nucleotide variation. We can have synonymous and non-synonymous mutations that affect or don't affect the coding sequences. It could be variation in the genomic regulatory sequences. So there's quite a number of genomic type of data that we could analyze. Epigenomics, which comes from the field of epigenetics, where we're interested in the regulation of transcription based on non-genomic variation. So microRNAs, long non-coding RNAs, different types of methods for pull-down techniques like immunoprecipitation, when we analyze ChIP-seq data, for example, or whole genome by sulfide sequencing. And then finally, transcriptomics. And so the objective of transcriptomics is to look at this continuum and close the circle, right? So as a proxy for protein expression, we can look at mRNA expression, looking at cDNA, right? So which is a copy that we've identified in the DNA region that actually is encoding the translated mRNA sequence. And so that's what we'll talk about in this program. How do we analyze mRNA expression using this cDNA proxy? And so to understand this a little bit better, let's kind of talk about the process. So as you remember, we're trying to analyze the expressed proteins. And so the expressed proteins come from translated messenger RNA. So in the process of transcription, the mRNA is produced. And then with the ribosome, we could capture those pieces of translated mRNAs, and we could sequence them to see what is going on. So what could be going on? What could be going on is we could identify the specific proteins that represent the activity of a cell or a property of a cell. So a property of a cell could refer to membrane proteins, those proteins that are maybe helping us determine what kind of a cell it is, what tissue it comes from, or what is its function. It could also show us what kind of signaling processes happen in the cell, or it could reveal subcellular processes that are happening at a given moment in time. So it could really tell us, what is the cell doing? What is its function? What is it maybe trying to do, right? And so as a result of that, we could understand the condition in a very precise molecular fashion. Many methods of RNA sequencing come from the microarray data analysis technique. So before this next generation sequencing became a thing, became kind of like an easy and accessible data type, there was first generation sequencing. And first generation sequencing really refers to sequencing using Sanger sequencing, where we have to annotate the whole genome and select specific regions to sequence. And it's a fairly complicated, labor-intensive and expensive process. And so before sequencing was applied to measure mRNA expression, people relied on other techniques like Northern blot or then came the microarrays. And so the microarrays were really invented in the 2000s. And we see how in the 2000s, those microarrays became very, very popular. And the reason why they became popular, as you can see right here, it is based on this hybridization technology and produces continuous data. Continuous data means that the visibility and the luminance of these dots really represents a continuum from 0 to 255, right? So you can really measure abundance of mRNA present in each cell. And so this is like a flow cell. Inside each cell, you have some specific transcripts that are complementary to the transcripts that are expected, just like you see right here. So in each one of these dots, you have these little pieces of mRNA. And then when mRNA from your sample gets put on this microarray, it lights up. And so as a result, you have this continuous data. Now, what led to the emergence of next generation sequencing? Two things. The first one is that we now understand that mRNA transcribed in the cell could be detected by looking at the genomic sequence where it came from. And so because we can identify exons, we know that those exons are transcribed into mRNA. We can then also start looking at real mRNA variation. And there's more variation to it than just abundance, which is what we can capture in the microarrays. So besides abundance, we can also look at what's called alternative splicing. And I'll talk in a little bit about the process of next generation sequencing, and you will see what are some of the important challenges that come as a result of this wider dynamic range. So the wider dynamic range here refers to ability to capture abundance as well as alternative splicing events and characterize this across multiple samples that represent conditions. So really it becomes a statistical problem to identify first where those reads are mapped to, from mapping to identify how they are distributed per sample, because we are talking about bulk RNA, so we're actually talking about sampling multiple cells, and then to infer from there, which is what you see right here, how that helps us differentiate or discriminate between different cell types. So here you can see that actually genes might have a lot of overlap because we are using here some kind of approximation. But when we are talking about this more detailed view of isoform and alternative splicing or exon-specific expression, we actually do have a lot of times a clear separation between conditions. So in this program, or what OmicsLogic is intended to provide you with, is an understanding of how these techniques actually fit in to conceptualize a process of biomedical discovery. And so biomedical discovery really is independent of the data type because it typically refers to this integration or association between the omics data type, which we will focus on, and the ability to describe the phenotype or characterize that condition using clinical data, chemoinformatics data, and other types of data sets that are used for diagnostic and therapeutic purposes. And so that is why this program is called Rna Seq for biomedical discovery. So before we go on, I wanted to make sure that this portion was clear. So if you have any questions, please let me know. You can put them in the chat box and we will be happy to answer them. I really want to make sure that we are moving along at a pace that everybody is comfortable with. And so if you need a little bit more time, or you feel like something was not clearly explained, please do let me know in the chat box. Now, I think to really appreciate how these methods could be used in real life or in the real world, we need to rely on projects that are sourced from important data sets that are available in the public domain. So why is it good as a resource? Okay, and Meenakshi here is asking, could you elaborate what you mean by abundance? Okay, so I will talk about that in just a second. But here you can see some examples of biomedical projects. So why we use this project-driven method to explain some of these technical, essentially, methods of analysis. The reason is because there is a true integration between computational side and biological side. You really have to be able to appreciate the biological problem at hand before you start choosing the methods of analysis and applying them to a data set that you have chosen. And so, for example, one of the things that we often include in this kind of a discussion is the problem of tumor and its microenvironment. So everybody has heard of cancer and the way that the tumor grows in a particular tissue. And more of the applications of this kind of analysis and biomedical applications have to do with the tumor microenvironment. Why? The tumor microenvironment represents a collection of a number of different types of cells, some of them immune cells, some of them are from the surrounding tissue, that could characterize in a much better way what is this tumor's property. So for example, it could help us understand the mixture of different types of cells, selection of immune cells, and characterization of their properties, but also then to understand how the tumor interacts with the tissue that it is surrounded by. So to talk about this challenge, we'll talk about Pdx models, which are patient-derived xenograft models, models that are mice, they're animals, but they have a human tumor that is engrafted in them. And because of that, using the simple mapping technique, we could differentiate between the microenvironment, which is essentially mouse tissue, and the tumor, which is human tissue. Okay, let me pause here real quick and answer these questions so that we can move on. What do I mean by mRNA abundance? Imagine a gene. That gene is being expressed, and this actually relates to this image right here. So here I have what's called a heat map. And in this heat map, I am trying to represent different genes. So here I have a list of genes, and here I have different categories or classes of samples. So when I have a gene, for example, ERBB2, that is highly expressed, and I know that it is typically associated with a particular type of samples, then I can look for whether that is upregulated or downregulated. So if it is expressed a lot, what could that tell me? That could tell me that a lot of the cells in a given tissue have that kind of an expression. And when I look at how I can target a particular pathway, and that pathway, let's say, is a signaling pathway, if I target ERBB2 or HER2, for example, I know that I have a targeted therapy that could affect that whole pathway, and that pathway essentially prevents this hypergrowth that we observe in tumors. So that is a good example of what I mean by abundance. I need to detect how many reads or the abundance of reads that map onto that genomic sequence of that particular gene to understand whether that is present and being expressed, and whether this target is a viable target in the context of that targeted therapy. That's just an example. Okay, Tripti was asking, can I elaborate the word chemoinformatics? So chemoinformatics refers to information about the chemical properties and structures in a given sample. So if we talk about a protein or a ligand, a chemical ligand, we can characterize those properties. We can look at atom properties, we can look at how they interact with each other, or how they interact with other types of atoms, for example, how they interact with other charged molecules, water, etc. So that refers to the word chemoinformatics when we look at chemical properties across different types of chemical structures and ligands. Okay, so can microarray differentiate between isotypes? I am not 100% sure what you mean by isotypes. Maybe you mean isoforms. So isoforms are transcripts. Genes is just the recorded gene sequence in the DNA. But when that gets produced, when that gets transcribed, there is what's called alternative splicing. So multiple exons are spliced together, sometimes in different order or in different combinations. And that is what we call an isoform. Essentially, it's an isoform of a protein, right? But we're using mRNA as a proxy for the protein. And so can microarray differentiate between them? Yes, only if it was designed to. So you will a lot of times see in the microarray data, instead of, how many of you, by the way, know how many genes are in the human genome? Maybe Vijay, you know how many genes are in the human genome? Just throw out a number. I mean, you can't go wrong. You're not graded on this. All right, there you go, 23,000, right? So somewhere based on different groups, it's somewhere between 20,000 and 30,000. It's about 22,500, right? So those are 23,000 is a good estimate. So when you look at microarray data, instead of 23,000, you actually see that it's like 60,000. So why is that? Because you can represent each gene by multiple transcripts. And there are some transcripts that are known transcripts. And we'll actually talk about this in a few minutes when we do a practical hands-on assignment. So you will see that a lot of times the microarray design includes known variation in order for a person who is analyzing that data to capture all the variation that is present. But since we don't know all of the possible variations, we are likely to miss some of them, because if we want to include all of the possible variations, it's just going to become too much of a, too many things to measure. So that's really the answer to your question. Final question, what do I mean by samples? So when I say samples, I actually mean a biological sample. When I take a tissue or a cell line and I extract RNA from that single sample, that becomes represented using digital data, and that digital data is associated with that sample, where it came from. Okay, so that's the biological sample. And actually, that's a good point to also expand upon when we talk about the need for normalization and incorporating that understanding in our downstream analysis. Okay, and Tejus, thank you for your other questions. Let me continue. I will make another pause in just a few minutes. We'll talk about other questions as well. So we talked about diagnostics and cancer biology, right? This is an example. Disease classification and patient stratification. What does that mean? So when you deal with a patient's population, we are saying that, okay, we could categorize patients using their gender, for example, their sex, male and female. We can talk about their age, their young age, 20s and 30s, old age, 50s, 60s, 70s. We can talk about comorbidities or clinical classification, where we say, okay, this person also has diabetes and heart disease, this person is obese, this person is healthy and running, right? So we can use different categories to classify patients. Now, this makes sense because we tend to think of people as objects that have these clinical characteristics. That's the clinical approach, right? You kind of look at the whole person and you say, this person has diabetes. But when it comes to selecting treatment, for example, for cancer, you don't essentially know what is the impact of these comorbidities or these clinical parameters on the actual tissue or cell types that you are treating. And so it makes sense to think about them as molecular combinations of tissues or cells that could be characterized using gene expression data. So you could take samples from these patients, for example, biopsy from their tumor, sequence them to understand variation at the gene expression level, and based on that, make decisions that have to do with signaling pathways or molecular characteristics of advanced or less advanced tissues and tumors. And so that's an important part, because ultimately using molecular characterization of patients or disease stratification, you can develop gene sets that are predictive of what will happen in a person based on treatment, based on disease progression, based on rate of recurrence, etc. So we will talk about that as well. And then finally, as we all know, taking a bulk RNA-seq sample is like measuring the temperature in a whole hospital, right? You will have people that are in the morgue, right, that are already dead, and you will have people that have acute infection and very high fever. And so if you average between the two, your average temperature might be normal. But in reality, that does not represent the different patients that are in your hospital. And so the same thing with tissues and cells. You can take a whole tissue or a whole sample, biological sample, but when you don't consider the individual cells and cell types, you lose a lot of the variability that can have a direct relevance to the type of biomedical problem that you are trying to solve. And so we'll talk about single cell and tissue decomposition, or in other words, how can we characterize the tissue based on the true variability that we observe in that biological sample. So ultimately, this whole program is designed to provide you with an overview of tools and resources for transcriptomic data analysis. It will provide you with an experience, and we'll do that, a little bit of that today, of how to process next generation sequencing data using what's called bioinformatics pipelines, and we'll use our tBioInfo server to do that. Then we will do analysis, normalization, and visualization of gene and isoform expression. We'll provide you with some automated tools. We'll talk a little bit about how to deal with this data in Excel, but we'll also show you some tools in R and Python that typically work as a package that you can learn how to use or modify so that you gain some insight into your data. We'll also talk about big data mining, machine learning, and predictive analysis. So what we talked about now, how to use classification, clustering, how to do predictive analysis on different types of complex data sets where you have hundreds of samples, maybe across many different types of variations. And so how do you go beyond statistical analysis to do that? We'll talk about analysis of single-cell RNA-seq data, how we can look for tissue-specific expression or cell-specific expression, which again has a very important significance for immunotherapy and all kinds of understanding of immune responses to different types of conditions. And we'll talk about different types of projects. So once you understand how these tools are used, how do you apply them in the context of your own personal project, right? And that project will be sourced from the public domain. So we'll introduce you to a number of different data sets that you can find on repositories like the National Center for Biotechnology Information, NCBI, Gene Expression Omnibus, how you can go to places like TCGA, the Cancer Genome Project, and others. So we will talk about a lot of different repositories and how to actually find a data set and start asking a research question about that data set. So the way that this program will work, so Velika already mentioned this, but I want to emphasize that we will have these regular sessions where we interact face-to-face like today. And before I talk about the other aspects, could I ask everyone to, just for a brief moment, turn on your camera so we can see who joined today. As you can see my face, I would love to know who else is here. So, all right, great to see you. Thank you very much. So again, the purpose of these interactive sessions is to be able to also meet each other and understand where everyone is coming from and what do you actually expect to gain out of this program. And so great, thank you everyone for turning on your cameras. We'll just give it a moment for the others to do that as well. So this program isn't just about me and about the participants we have here, it's also about you. And so we would love to get to know you in the process of this learning experience. We would love to understand what you are currently working on. If you're a researcher, a student, perhaps you are trying to apply some of these methods to a data set that you are interested in. And so, you know, that is another big part of this program, is to really hear from you and understand, understand where this is coming from. Okay, so I will save this screenshot. It's wonderful to see all of you. And if you want to turn your cameras off now, that is fine. And hopefully in this program, we will be able to get to know you a little bit better. So let me describe now how this program will actually work. So this is the link that we have all of the program sessions organized in. You can actually go to learn. Let me actually paste this link in the chat box. Okay, so on this link, you will find this program. And by the way, if you're already registered on the Learn portal, you can just click on Programs, and you will see the programs right here. These are the programs that we have, but this is the program that we're going to be looking at today. So once you're logged in and you're a member of the program, you will see the overview as well as courses. You can book a meeting with our team. So you can actually schedule a one-on-one mentor session if you have the subscription for that. There is a section here for our Slack channel. And so those of you that already registered, you can go to Ask Question and you will be able to join our Slack channel. So I'll just show you how that works. So here I have the people joining the Slack channel, and I can actually post a question. So for example, I can write question, how do I find a data set on Rna Seq? Okay. So now you will find this appear right here. So this allows us to communicate back and forth, and that's the objective of this program. We don't just give you some resources and tools to use, but we also can have a discussion. So if I now reply to this, if I reply to this, there we go. You can go on, okay, something like this. So that will now appear here as well, right? So that way we can have a discussion about these topics. If you don't understand something, if you have questions about something, you are free to post that right here. And importantly, in between the sessions that we have in this interactive way, there are some additional resources that you have the freedom to go on your own pace. So that's an important part of it. We understand that many people have a very different pace of learning. You can learn by just attending sessions and asking questions and learning about the hands-on exercises that we do together, or you can learn more technical things by completing this coursework. And so we have some introductory courses. We have some comprehensive training on transcriptomics. And then you will see that we have also some project examples that you can apply the methods that you learn to a particular data set. This is a data set on the TCGA, the Cancer Genome Atlas. We also have some courses that you can practice coding, for example, in Python and R. And we'll add some more projects here as we go along the way. So let's take a look at some of the topics that we will cover. But before we do that, let me know if anyone has any questions about this page. Again, some of you might be seeing a different view, and that is because you're not registered for the program. So just to kind of illustrate what that looks like, if I go to the program page, and I see this when I'm not logged in, so I'm not a part of this program, you can see that just the information about this program is available. So you can see the sessions, you can see the schedule of these sessions, you can see some of the things that are being offered and some of these testimonials. And if you want to complete your registration, you can complete your registration using one of these options. Beginner, which does not give you access to one-on-one mentorship. Intermediate, that provides you with one-on-one mentor support. And advanced, that also gives you extra time to work on a research project and gives you access to some of the big data analysis tools that we will be using throughout this program. So if you have any questions, think about those questions and let us know in chat or follow up by email later on. Okay, so Mohit here is mentioning that it would be great if the syntax is explained in the coding exercises. So I will get to that in just a few moments. I'll give you an example. I'll give you an example of some of the exercises where the coding syntax is explained, but we will talk about that. And thank you for that comment. All right, so let's go back to Rna Seq. One example that I want to show you is an example from a real data set that we will be analyzing using the big data analysis platform so that you can appreciate the logic of Rna Seq analysis. So how is the data mapped? how is it then analyzed for variation using comparisons between different conditions. So if you could please, again, once you're registered on the Learn platform, so once you're registered here, the same password will be used for this big data analysis platform. So I'm going to give you just a couple of minutes. If you could follow to this link, again, you have to understand you have to use the same password. And please let me know once you're here by putting in something in the chat box so that I know that you're here. And I'll give you just a couple of minutes. So in the meantime, I'll explain what this is. So our company is essentially a tech transfer company. So we work with a bioinformatics research center at the University of Haifa in Israel. That center is focused on computational tool development. So they're really focused on making bioinformatics accessible to anyone with or without a background in bioinformatics and with or without engineering and programming training. So it is for biologists and for clinicians. Okay, and I'm going to continue talking about the platform until I start seeing that some of you are already here and have logged in. So please, once you are here and logged in, put in something that's in the chat so I know how many people are following along. So one of the projects that they have been working on for the past several years is this big data analysis and integration platform. And the idea behind this platform is to offer tools that are easy to use across several different types of omics data, like next generation sequencing, mass spectroscopy, structural biology, as well as tools for data integration and modeling. In this program, we're not going to be able to cover all of these tools, but we're going to rely on a number of these tools to help us appreciate the process of analysis of RNA-seq data, some of the introductory data mining tools, and then data visualization and biological interpretation. All right, great. So I see that 1, 2, 3, 4, 5 people are here. Let's give it just a few more moments until everybody else is here. Now, you need to be here and you need to be logged in. How do you know that you're logged in? You know that you're logged in if you hover over this right here. You will see your email right here. Okay, so 1, 2, 3, 4, 5, 6. All right, 2 more minutes and then we will go on. So the way that we use this in the context of our training is, first of all, we're going to show you some demos. In these demos, you understand the logic. How do the different steps connect to make a pipeline, to make a working analysis workflow? And then once you are able to replicate those demos and you understand all the inputs, outputs, and the steps in between, you will then be able to analyze your own data. So then we'll transition into a more hands-on practical analysis of your own data or data found on the public domain. So to get a sense of what that looks like, let's go right here. You can see transcriptomics, and the first section in transcriptomics is basic processing of transcriptomic data. So let's go into this first demo, Rna Seq cell line project. Okay, how many of you already have some experience in Rna Seq data processing and analysis? Is there anyone here that has already bioinformatics experience? Okay, Avinash is saying that he doesn't have that experience yet. Okay, Kibgot Potso is saying that yes, he does. That's great. So if you don't have any background in Rna Seq data analysis, this will be a little bit new and you might want to repeat this again on your own. For those of you that already have that kind of an experience, I want to kind of also explain why this is a useful interface for you to appreciate the different methods that have developed over time and how they compare to each other so that you can make comparative analysis of these processing steps. First of all, let's click on the first button called Start. Here we have the three main steps that this pipeline offers us to do. Pre-processing. So when we generate those digital reads, and those reads are in abundance, we know that they have been prepared using library preparation techniques. In other words, there's adapters, short sequences that are used when those reads are generated. So that's problem number one. Problem number two, the amount of reads has been amplified using PCR. And so because of that bridge amplification, there is a difference in abundance between different types of sequences, because we know that PCR is just biased. Some sequences are typically more amplified than others just because of the type of sequences that they are. And so that represents some technical variation that is not wanted, right? It is going to skew our results, and it might have an impact on our biological interpretation. Now, this is especially important when you deal with genomic data, because you might be finding variants because of those adapters and not because of the true variants found in genomic data. But in transcriptomic data, this is important because some of the highly expressed genes tend to be affected a lot more by these PCR amplification techniques. And so it's important to eliminate that technical variation. The second part is mapping. What is the challenge with mapping? Can anyone explain to me, maybe just in 2 seconds, 2 sentences in chat, why are there so many tools for mapping of these reads onto the reference genome? I mean, what's the problem? You take a read and you place it somewhere on the genome. Why is this such a big area of bioinformatics tools, and how come we have so many of them out there? Okay, I'll give you another 2 minutes to think about it. Why do we have so many tools? Let me just close this and I'll show you what I mean by many tools. So here we have a mapping section. All of this section refers to mapping. We have here star, BSQLB, bow tie, BWA, top hat, high set. Okay, so Zara is saying, I think not full coverage of genes sequencing. So I'm not sure exactly what you mean, Zara, but okay, Favor is also saying because there are many transcripts and we need to find the right positions of the transcripts before making use of the gene info. Okay, so yes, the number of genes, the number of possible positions. So to appreciate why we need so many, okay, we need to actually appreciate how this data gets generated, right? So let's briefly take a look. This is the session where we talk about how data is generated in next generation sequencing. So let's just talk a little bit about what that is, right? So here we have, here we have a complete pipeline. And when you start, you essentially load the data into your pipeline. That data could be of many different formats and many different types of sequencing. So you have different protocols, you have different types of sequencing like pair-end and single-end. You also have different types of read lengths, and you have different amount of reads for the whole sample. So you can have millions of reads, but you can have 10 million, 5 million, 3 million, right? So different coverage. So pair-end and single-end, different methods of data preparation using different library techniques, library preparation techniques, different sequencers that produce different lengths, and ultimately you have also different genomes. Now, how long is the human genome? Can somebody tell me? How many nucleotides are in the human genome? Okay, perfect. 3 billion, 3.2, yep. About 3.2 billion nucleotides. How long is a read? Does anyone know how long is a typical read sequence? What's the length? Okay, 151, 3. The truth is, it's between 30 nucleotides and about 350. There are some long read sequencers that now sequence up to maybe thousands of base pairs, but typically we're going to be dealing with anywhere between 50 and 150. So let's take a look. This is a pipeline for a complete RNA-seq analysis, but let's build a pipeline to just understand the read mapping part of it. So we'll click on Start. We'll go to Bowtie 2G, which is one of those mapping techniques onto the human genome, which, as we know, is 3.2 billion base pairs. So we take a short read, 50 base pairs, and have to find a place on a 3.2 billion length. Okay, so we'll click on OK. And then we'll click on visualization. So we'll see how do those reads actually map onto this reference genome. I'll click on End. Okay, you can follow along, but you don't have to. I'm just going to show you the output of this pipeline. Okay, and we'll take a look at what is actually going on. So these are individual reads. And as you can see, they have been mapped onto this reference genome. And if I click on this read, okay, first of all, you can see that they are red and blue. Okay, how long is this sequence? How long is the sequence of this one read? $150. Okay, thank you, Favor. Right, you can see right here, sequence length 150. So 150 letters have to be mapped onto this long reference sequence. Not only that, you can see that some of them are read forward and some of them are blue backward. So they are complementary sequences. They go in two directions. That gives us higher confidence in how the mapping is done, but it represents quite a significant technological challenge. And so when you think about the applications of genomics or RNA-seq data analysis tools, those are two very important trends. The development of sequencing technologies, which led to longer reads, more accurate reads, and cheaper reads. And at the same time, these computational tools that started to develop analysis methods for quick mapping. Okay, so let's go back. Okay, let's go back to our pipeline and see what are some of the other methods that we have here, right? So we have Bowtie 2G. This is just mapping onto the reference genome. And we know that there's 3.2 billion positions on that genome. We could, in an alternative way, map onto the transcriptome. What is the transcriptome? Out of the 3.2 billion, only 1% are actual coding sequences. So what is 1% from 3.2 billion? Mathematicians out there. And I see here somebody is asking for the link. So we're actually just using this pipeline right here. Okay, who can calculate 1% of 3.2 billion? Okay, Tejas, sorry, I don't think that, yeah, people outside of India actually know what lakh means, so neither do I. I think it's something like 100,000, but I'm not sure. Okay, Mohit is saying 32 million. So what is a billion? Okay, I won't torture you, but you can appreciate now the difference between millions of positions and billions, right? 3.2 billion. So there is a difference of, there is a huge difference in how many positions we have to survey to find the best mapping place for a short read. And so instead of using Bowtie 2G, let's start another pipeline to see what that would look like. We can actually map our data onto the transcriptome. Okay, but before we do that, let's pre-process our data. We'll eliminate the adapters, clean up PCR clean, and we'll learn in our next session about this PCR bridge amplification. Then we will add the reads, map them onto the transcriptome, or all of the expressed genes, all of the exons essentially that are present in transcripts. And then we have to create a table, right? A data frame. We need to create a table of expression. And so when we create that table of expression, we're also thinking about how that table represents the statistical complexity of the data set that we have here. What is the complexity? The complexity is that genes are of different lengths, and there's different number of reads for each sample. And so when we look at preparing a table that we want to use for downstream analysis, there are some normalization techniques that we have to learn about to normalize for read length, or sorry, for gene length, and for the total abundance of reads available. So that's why we use different methods like FPKM and TPM. And now when we complete our pipeline, we will see here a number of outputs like the expression of genes, exons, and isoforms. Okay, for TRPT, how do you access this screen? You go to server.tbioinfo, right? So again, this is the link. Just gonna put it here. So if you go to this link, you have to sign in and use the same account information like you created on Learn, okay? Okay, so this gives you a snapshot of why we use this tool. This tool is easy to combine different methods, different steps together in a workflow, like a pipeline. It's reproducible. I can remember what I did. It is color-coded, so you know that all the blue is pre-processing, all the red is mapping, all the green is quantification. And you can also perform both data processing, so like we did now, or you can use also comparative analysis, so use some statistical tests to identify genes that are differentially expressed. Okay, so this was just a brief example. Does anyone have any questions about this example that we did? Okay, if you do have questions, put them in the chat box. We'll run some pipelines. We'll understand the logic behind analysis. We'll be able to see some outputs that are both visual and just as data sets. So this is a volcano plot. We'll talk about what does it mean and how to use it. And we'll talk about biological interpretation. How do we actually understand the meaning, the biological meaning of thousands of genes that we potentially have in a given sample or in a given differentially expressed pipeline. And then we'll talk about how do we account for the variation in our source samples, in the biological samples, and what can we do to explore and normalize this data, as well as explore this data to inform a data-driven research question. We have a number of courses that combine statistical analysis methods with an understanding of complex gene expression patterns, normalization techniques that involve log-scale transformation, and we'll also talk about data mining and classification. Well, we'll talk about different methods of big data analysis that are commonly used to cluster and classify such data. So then we will talk about how this applies to specific biological examples, like the ones I mentioned in the beginning. In cancer, we have different subtypes of cancer. How do we understand their differences, and how can we ask questions about those differences? And then we'll introduce you to some coding exercises where we will explain how to load this data, visualize this data, and also how to perform some similar analysis steps using coding. So that coding will be done using our Code OmicsLogic playground. So let's take an example of one of those exercises. Okay, TRPT, after you log in, maybe the best thing would be to actually view this recording that we will share with you and try to do that there. Okay, so let's take a look at an example of a coding exercise that we will do in this course. Okay, so what I'll do here, this is on the course page. So if you're already here, I'll just give you one example of how we will load and visualize this data using R. So here's an example. Let's go to data visualization. Okay, so when you deal with data, different types of data go with different methods of analysis. So you can have maybe information about your input data, about the different samples that you have. That typically will be categorical data. Categorical data means I have 5 samples that are ER positive, 5 samples that are triple negative, 5 samples that are HER2 positive, something like this, right? So it's categories. There's no continuum between them. There could be continuous data, and continuous data means that I have gene expression anywhere between 0 all the way up until hundreds of thousands, and I have all the possible ranges in between. There's no major breaks. There's not just like a lot of 5s and a lot of 10s and a lot of 100s, right? It's just continuously a continuous scale. And so for the different types of data, you can use different visualization techniques. But before you actually visualize the data, you need to learn how to load it and how to understand what's in it, because your data frame that you load can have numerical data in a matrix. It can have some headers that explain what each sample name is. It could have some row names that, you know, specify gene names. So we have to be able to prepare our data before we actually visualize it. And so what these steps do is they actually provide you with an explanation and then offer you some practicing steps that you can do on your own. So you can actually practice this right in your browser. Let me share the link right here. Yes, Vijay, we will definitely provide you with some data sets, and those will be introduced in the context of that project. Okay, so here we are loading some data. You can see that I'm creating a data frame. So first of all, it's just an object, right? R is an object-oriented language, so we create an object, and we add here a data table. So this data table, you can look at what does this table actually look like. So let's copy this link, and we will put it right here. This is what the data looks like. There are some names on top. This is the header, and there's some genes right here. And for simplicity, they're called gene 1, 2, 3, etc. And in between, we have some numbers. So we can go in here and we can load this table. And we're saying here, skip 1. Okay, so we are going to be using, this code is an R. Okay, so you can see here that we are loading this data. We're then creating a data frame. We're removing some of the numeric data, creating a matrix, okay, and we can then check the mean. So we have the mean for our data. So what's the average or what's the mean for our expression values, okay? And so this is a very simple step. You can do this exercise, and this exercise is for a given data set. Now, I can change this data set, right? I can take another data set and check the same right here. Now, how to visualize this data? Again, you can just simply run this code, okay? And you can see that there are several types of plots that you can create. What is this called? This is called a box plot or a box and whisker plot because it represents the interquartile range, maximum and minimum, as well as outliers that are present in each sample. So here you can see sample names. Here you can see a complex visualization called the heat map, where you also cluster your genes and your samples together to represent the variation by groups. Or you can look at the distribution of data, and you can see that there's maybe something that is not normal, right, about this data. For example, here you have a lot more low expressed genes than what you would expect from a normal distribution. So now you can modify this. For example, if I change this to purple, okay, you can see that now this is purple. This is actually going to work, and I can modify this. And the objective of the future steps and exercises that we will do is to take some practical coding exercises like these, going from very simple to more and more advanced up until you're able to write your own code. So as you go through these exercises, you will go from very simple, just press a button and change a little thing, to eventually completing whole sections of code so that you understand how to write code on your own. There are also going to be instructions on how to download and install R and Python on your own computer, on your own machine, because ultimately this online environment is going to give you some limited expertise, but you have to set up your own environment where you have your own files, you can install your own libraries, you can explore different tools that are out there in the public domain. Okay, let me finish with one last thing. I see that Vijay was asking, how do I find some data? Where do I find some data that I can practice with? Okay, so Harsh, why are we converting the data frame into a matrix? There are different methods. Some methods only work with numeric data, which is a matrix, and some methods know how to handle headers in a combined data frame. And so based on the different methods that we use, we either have to prepare a matrix first, or we can use a full data frame, but sometimes we need to transform it into a different format. So we'll talk about these formats in our sessions. If you copy the file location details and paste, but the feedback says the file doesn't exist, check the link. If the link opens and it does exist, then it should work. Yes, so Meenakshi, we have a platform. So we have a server, right? So we're using a server. The server runs an R environment that is handled by our server. So that way, we don't need to go through the steps of everybody installing R, making sure you have all of the different packages and dependencies and things like that. So this is just a simple way for us to all do the same steps together. All right, so where do you find these data sets, and where do we find the projects that you're going to be doing in this program? You find these projects, for example, on a place called NCBI, the National Center for Biotechnology Information. So I'll just paste the link right here. Okay, can anyone here provide me with a topic, a biomedical? Let's think about some biomedical topics. Yes, I will show you how to download some data sets from here, but somebody give me a topic. What are you interested in? Some kind of a, let's say, liver cancer, diabetes, okay, SARS-CoV-2, perfect. So let's look for SARS-CoV-2. Okay, SARS-CoV-2, obviously there's a lot of different types of information about SARS-CoV-2. So when you scroll down right here, you see a lot of different types of data. Now, for our purposes, for transcriptomic data analysis, there are going to be two repositories that are going to be most interesting. The first one is GEO, which stands for Gene Expression Omnibus. Okay, so these are typically already tables of gene expression that you can just download and analyze. Or SRA, which is Sequence Read Archive. And so remember the pipeline that we built to actually process the individual reads and map them onto the reference genome to create a table of expression? That's what we would use here. Now you can see that there's a lot more SRA than there are GEO. And the reason is because SRAs combine individual samples. Each SRA is an individual sample. And here in the GEO datasets, they're grouped together. But also not all of the SRA datasets that are available on here are already processed and available for you to download. So here we can take a look at some of the existing datasets that we can just download. And for example, you can see here over 2,900 samples, right? 1,702 samples. So there's a lot of samples already here, 784, right? 493, 407, 363. Sorry, a lot of samples. Let's find something with less samples. Okay, these look like individual samples. Okay, and so here you can also customize. You can say, I want to look at high throughput sequencing and change it to that. And also we can customize it by, yeah, there's just huge samples, right? Sample sets. But you can see that you can easily download them, and obviously you can, explore them on your computer. So just to give you an example, 96 sample, okay, 88 samples right here. So you can just download this data. Okay, and you can see that it comes in many different formats. For example, raw counts, which you can use for differential gene expression, normalized counts, okay, and then log transform count. So let's just download raw counts, and I'll show you what that table looks like. Okay, so here you have a data set. Let's open it real quick in Excel. We'll look at what it contains. Again, I'll share this link with all of you. You want to download it for yourself. All right, so we can see here the ID. So there are some lung, heart, and other tissues, and you can see the name. For example, let's take a look at IL-33, which is interleukin 33, a gene responsible for inflammation. And we can look at, okay, what is its expression across the different cell types that you have, right? So here you have some variation. It is highly expressed in just a few samples, and typically it's low expressed. So this kind of a cytokine is typically expressed when the cell gets infected and actually dies from infection. So now you can appreciate how this type of information could be useful to differentiate between different samples that we have in this data set. All right, so thank you for listening to my part of the presentation. I will now pass this on to Dr. Mohit Mazumder, which will be one of the mentors who will help you develop projects and guide you through the steps of analyzing your data, and also will be able to show you about how the training registration process will work and what are some of the upcoming opportunities for us to learn about different types of omics data in the context of this program. Over to you, Mohit.